All right, so actually it is really a pleasure to welcome here Kresten Lindorff-Larsen, and it's fantastic that he agreed to come and give the seminar for us for the TYC, and I'm really happy to see so many people who are here. So this is great because this is now our biomolecular simulations or maybe condensed matter interest group seminars, really the first one that we have started now after this COVID. And although it's still online, but luckily due to the tube strike, I think it's not a problem. And so it is really great that Kresten is here. And as I've mentioned before, now just for the recording, I would like to thank again Alice, who have initiated this invitation. And I would really like to ask if any of you have some suggestions for speakers, please contact me, please let me know. And I'll be very happy to try to get some fantastic speakers like Kresten. Hopefully they will also agree. So just a little bit of background for Kresten. So he got his PhD at the University of Cambridge, working with Professor Chris Dobson, which is an experimental group, but he has already done molecular simulations at that time. And then he became an assistant professor in Copenhagen. But after, he joined the D-Show research group, where he did really some fantastic work. And so what is also nice to see is that from that kind of environment, he nevertheless came back to academia, which I think is more of a rare situation nowadays, unfortunately. And so he is now a professor in computational protein biophysics in Denmark. So he also received many different awards. And for example, he was co-recipient of the Gordon Bell Prize. And I think I don't even want to waste too much time on me trying to introduce him, because I think most of you really know his work. And I think all of us are looking forward to his talk on intrinsically disordered proteins. So thank you, Kresten.
Thank you very much, Idina, for that introduction. And thanks for having me online or not. It's always my pleasure to speak, if not to people, at least with people. So I'm also happy that I've already got the chance to speak to several people, and look forward to speak to more people also. I just need to find the right buttons to click here. Although first I should minimize this, sorry. And then I will do this. And unless someone complains to me, I'm assuming that you can see my slides and stuff. So as Idina mentioned, I started out my life as being an experimentalist, and I still collaborate a lot with people that do experiments. And so this is sort of, I think, our main angle into working with simulations is we try as much as possible to interface our simulations with experiments in various ways, sometimes indirectly and sometimes, as I'll hopefully give you a couple of examples of, very directly. And I'll give examples from the world of intrinsically disordered and flexible proteins as examples, because I think that they are perhaps the kinds of proteins where this is most needed. But I think many of the things that I'll talk about today will also be relevant if you're working not on these kind of highly disordered, complicated, floppy proteins. I work at the Department of Biology, and so many of my colleagues, range from being protein chemists to people that study, let's say, real biology. And so I like to lead off by showing a picture of an undeniable biological object, a horse, but soon we'll be switching into molecules. But the idea here is that I just want to remind you, and of course, for those of you who do modeling, maybe I don't need to remind you why we do simulations and not just experiments. But the basic idea is that while experiments can do a lot, they can't do everything. So if you, for example, are interested in a process, like in this case, how a horse gets from one place to another place, if you have a well-behaved system and you're a good experimentalist, you can sometimes get experimental data that looks like this, where you have both high spatial resolution, so you can see what the horse looks like, but you also have high temporal resolution, in this case via using flash photography. So you actually record a series of snapshots of some process as it's taking place. So you can sort of get information maybe about the stable state over here and maybe a stable state over here, but also get information about what sort of the transition state structure of this process would look like But most typically, you don't actually get this kind of information. Instead, you get information that looks like this. This is the same technique as before, and this is a picture I try to show in as many talks as I can. So same technique, photography, but quite different. Not only is it not a horse, but a swimming pool, but it also looks in some ways a little bit less interesting, at least at a first look. But actually, when I first saw this picture, I was extremely fascinated by it, and I still am. So what struck me at the time, and maybe strikes you now, is that the lighting is sort of weird, and one of the things that I realized at the time was that there was sort of something odd about the light, and if you were in the same room, we would discuss it in more detail, but you would sort of notice that there's light coming in both from this side and from this side, although this is of course the sunlight, and there's only one sun present in our solar system. And the reason for this is that while this is again photography, the exposure time or the averaging time of the experiment is very different from the previous picture. So this is a picture taken with a pinhole camera with an exposure time of about a day, so a very small hole that lets in just a few photons at the time, but then compensated with a very long exposure time. And so if you're doing an experiment on a system while it's moving, you get these kind of artifacts. So for example, that it looks like that there's two light sources when in fact it's just averaged over a very long period of time. And also you don't really see the people that were actually swimming back and forth in these lanes during the recording of this experimental data set. So what you see now is just some incredibly flat water, of course completely unrealistic, but this is what you get if your experiment, and now I'm sort of starting to get into the world of molecules, if your experiment has an averaging time that's much longer than the timescales of the dynamics of the system you're interested in. So if you're interested in just what does the average swimming pool look like, this is a great experiment. If what you care about is how does the sun move across the sky, how do people swim back and forth in lanes, and what are the waves on the swimming pool, then this experiment tells you something, but it doesn't tell you a lot of the details, right? It tells you what the ensemble or time average should look like, but it doesn't actually tell you about the dynamics of the system. So we do simulations because we care about these dynamical processes, and because most of the structural biology experiments that are available, they don't directly probe the dynamical motions of proteins. They just give us a sort of ensemble and time average view of what the systems look like. So today I will tell you about three different things. And I will be spending more and more time as we progress through these things about how we can use experimental data together with simulations. I'll just give you a single snapshot of some methods that we have been using quite a bit and have developed some software to do when we can sort of use experimental data to refine simulation models in the sense of some system-specific corrections. And then I'll talk about two things that we've done more recently on how to improve simulation models, in particular force fields, coarse-grained force fields for disordered proteins, by essentially benchmarking them against experiments, or at the end really optimizing the force fields directly targeting experimental data. So this is sort of the outline of the talk. And I won't dwell too much on this, and this is something that probably you are all familiar with, but when we talk about the relationship between experiments and simulations, we sort of, not completely hard, but sort of divide the methods into sort of two approaches. Either we run simulations, we have a lot of coffee here, we have good coffee machines, and while the simulations are running, we have coffee and we start thinking, and then eventually we use what we call forward calculus. We try to predict some experimental observables, and then we can sort of benchmark the simulations. Do they agree with experiments or not? But what we can also do, and what we and many other people also do, is sort of what we call inverse modeling, which is in many ways similar, but in some ways conceptually different, is that we have some set of experimental data we need to interpret, and then we use the simulations as an interpretation tool by explicitly constructing structural models that fit the experimental data. So instead of using the experimental data for benchmarking, we sort of try to use it as a model that we can sort of massage to fit the experimental data. These two approaches are different, but they are not sort of completely independent from one another. So if you benchmark your simulations against experimental data, you will eventually get better and better models. This can be better force fields or other prediction methods. And the better model you have, right, so this is effectively sort of moving on the left on this axis here, the more you can do just using the computational model on its own in the absence of experimental data. But what it also means that if you have some experimental data available, then you can integrate them and you can say more with less data. So if you have a pretty bad computational model, let's say, you know bond lengths and bond angles, and maybe that atoms shouldn't lie on top of each other. This is sort of what we typically use when we do normal structure determination. If you can supplement that with high resolution data from crystallography and MR-EM, you can determine beautiful structures at very high precision and accuracy with a pretty simple computational model. If you have a very advanced computational model and no experimental data beyond maybe the protein sequence, you can, in favorable cases, predict the structure either with a physics-based model or with some more advanced machine learning-based model, and that also works. And then there's all this stuff in the middle that we and many other people like to play around with, where you have some kind of computational model. You know that it's good, but it's not necessarily perfect. So if you just use simulations or computational methods alone, you won't get the right answer, but you will get a reasonable answer, and then you can sort of massage it with sort of low resolution data of different kinds. And so pushing the accuracy of force fields and also sampling methods sort of to the left allows you to study more complicated systems with less and less data. And so that's why we sort of, even though these methods are to some extent independent, they are also sort of useful for one another. So for those of you who have done simulations on cases where you can also measure things experimentally, you will often find that, that your simulations results do not match perfectly the experimental data that you have available. And there are many things that you can do. One thing that is often entirely reasonable to do is that you publish a paper saying, we did this and it doesn't match perfectly. We think, this could be the reasons, and then you move on. So this is something, we've done that. I won't talk about that. And then another thing that I'll talk briefly about is that you can sort of combine the two and refine the computational ensembles using your experimental data. And I'll tell you about how we do that in a second. There are other ways of doing it. And this is sort of a system-specific correction. We find that to be extremely powerful for understanding specific systems. But of course, also, if your simulation is converged and you're interpreting the data in the right way and it doesn't agree with the experiments, and the experiment was also was done in the right way, then it's, at the end, it tells you that your force field is wrong. And so what I'll talk about at the end is, how we can use this kind of information to drive the improvement of force fields. So this, I know, is quite vague at the moment, but hopefully it will become a little bit more concrete as we get to the different parts. And again, I just want to highlight this important thing with nearly all experiments is that there are ensemble average data, and you need to remember this when you're comparing your computational models to the experimental data. This is sort of my simple one-slide summary of NMR spectroscopy. We work a lot with NMR spectroscopists here. And so NMR spectroscopy is something with spins. So imagine now that I'm collaborating with someone who studies the structure of coins, and they can attach this sort of vector to the coins, and they can measure whether it's spin up or spin down. And they come tell me, yeah, Kresten, I studied this coin, and I measured the Z component of the vector, and the value is 0. Can you tell me the structure of the coin? And the standard, let's say, way that people do structural biology is that they would take the coin, they would rotate it, and then they would find a structure that agrees with the experiment, and this would then be the structure shown here at the bottom right. This has perfect agreement with the experiment, that the Z component of this vector is 0, and this is what was measured experimentally. And of course, you realize with this silly example that this is not correct. And the mistake that I made was that I interpreted an ensemble and time average experiment as something that should be satisfied by a single structure of a coin. And in this case, you get a sort of almost nonsense result. But of course, in the context of biomolecules, you can also get all sorts of, also semi-nonsensical results. And so, of course, the true answer here would be that, a 50-50% mixture of spin up and spin down would also give you a 0%, a zero vector component. And so this is something that we need to remember when we're interpreting time and ensemble average data is that we cannot just look for individual structures that agree with the data. And the more dynamic the system is, the more important this becomes. So this, realizing now that nearly all structures in the protein data bank are determined ignoring this thing, right? So there is really determined one set of X, Y, Z components of all the experimental data that essentially explain the experimental data. And this works when the proteins are not very mobile. But for the more mobile proteins or data that reports on mobility, this eventually becomes completely meaningless. So instead, what we do is that we use molecular simulations to generate not just single structures, but ensembles of structures. And then we somehow need to combine them with the experimental data. And this is a field that I and many other people have been working on for many years. We wrote this review summarizing some of these ideas, and I can recommend, for those of you who care about the technical details, I can recommend this review to look into sort of an overview of things. And there are many different approaches, and they can be split in many different ways. But essentially, the basic idea, if you have some kind of force field that you can sample that gives some kind of distribution, or in this case, where I'm plotting on the energies here, you can integrate it with experimental data either during the sampling. So this is sort of this path up here. You can somehow plug in the experimental information, and you can sort of reshape the force field so that when you start sampling the force field, you get out distribution of observables that agree with experiments. This is called biased samplings, where you're having experimental bias in the sampling. This can be extremely powerful. Technically, sometimes a little bit difficult how to do this. So this is one way. This is not what I'll talk about today. The other thing that you can do is that you can take your force field that that doesn't know anything about experimental data. You can sample it. You can get out another distribution of observables that in this case would not agree with the experiments, and then you can add in the experimental data in some process that we would call reweighting that I'll talk about in a second, where you sort of change this probability density function of this ensemble in some space to massage it into agreement with experimental data. So you're using the computational model as an interpretation tool rather than a prediction tool. And the approach that we use is a method that we call Bayesian maximum entropy ensemble refinement. The basic ideas are developed not only by us, but by many other people, in particular Gerhard Hummer and Giovanni Bussi, Michele Vanduscolo, John Cordero, and many other people have worked on this for a number of years. In my group, it was particularly Sandro Portato, who developed some nice software that's available at this link here and described in some of these papers. And you can formulate it in many different ways, either from the Bayesian side or the maximum entropy side, and it sort of ends up being more or less the same. And the basic idea is explained in this figure here. If you have some kind of force field, this would be this red density distribution over some quantity that you can calculate, measure the average of, and you get sort of this average here indicated by this red line here. And then you go to the experimental world and you measure some number. This would be this black dashed line over here, maybe with some error bar. You can say, okay, the number that we measure is not in agreement with the experiment. How do we massage the system to become an experiment? We perturb the system, or we reweight the system. We change the weights of the different frames in our MD with some minimal perturbation. And this is where the entropy side comes in. So this is a minimal cross entropy or maximum entropy approach, where we sort of decrease maybe the population of this state a little bit and increase the population of this state a little bit. In this way, we sort of move the average from this wrong value to be close to the experimental number over here. And of course, we can do this not just along one dimension, but you're targeting many different experimental observables at the same time. And there's a lot of details that I'm sort of neglecting here, but this is sort of the basic idea. You can formulate it in many different ways. One way that I like to think about it, in particular, for teaching this to people who are more thinking about it, thermodynamics, is that we have sort of a function here, gamma, that we call gamma because it sounds a little bit like a Gibbs free energy, and we want to minimize this Gibbs free energy, and it consists of two terms, a chi-square term and an entropy term. The chi-square term measures the deviation between experiment and simulation, and it's a function of these weights that I'll talk about in a second. So we want to minimize this, but at the same time we want also to not sacrifice the entropy too much, and in this case the entropy quantifies the deviation from the sampling we had with our force field. So this would be in the Bayesian framework, what we call our statistical prior. So we want to minimize this function here, gamma, by improving agreement with experiments or minimizing the chi-square. without having too big a reduction in the entropy. And of course, like in the thermodynamic sense, there's a balance between the sort of the enthalpy and the entropy kind of term. That's this theta parameter that plays the role of a temperature that sort of sets the balance between these two terms. And this is an important parameter to set, and we can discuss that later. And these are methods again developed by in particular Gerhard Hummer and as I mentioned previously, many other people. And so what we do is that we have a set of frames of an MD simulation. Each of them have a weight that might be uniform if we just do normal sampling by MD or Monte Carlo. And then we optimize these weights. So we change the weight of each frame in the MD simulation a little bit to improve agreement with experimental data while not sacrificing too much on the entropy side. So how does this look? Again, a toy example just to illustrate the idea. If you have some force field, this would be the red line over here that is not perfect, but is a reasonable force field. And in this case, we can sort of pretend that we have a true force field. This is the one that we wouldn't know. This would be nature's force field. This would be the gray 2D histogram here, or the marginals shown here at the top and on the right. And so you can see that there's not the force field that we have available doesn't agree exactly with nature's force field, but it's not completely crazy. So for example, if we calculate the average of X and Y, with the force we have, this is this number here, this is this star here. Nature's force field would give us this, black point here. If we go make a measurement, we don't always get, the exact truth. So this would be the measurement. So what we would then do is that we would optimize the weights of our MD simulation so that we would, essentially move the calculated average as close as possible within error to the experimental number that we measured over here by changing the weight of each frame, by minimizing this function gamma over here. So one way of visualizing this is that we move along this gray line here. And as we're moving along this gray line here, we are changing this temperature-like parameter. So when the temperature is very, very high, we put all our knowledge on the prior or on the entropy. We essentially want to sort of not change the prior. So we end up with a high deviation to experiment, but a high entropy, and this we can quantify effectively by the exponential of the entropy is the fraction of the MD frames that we sort of keep in the simulation. So this is essentially very high. But then what we see is that by changing this temperature, decreasing the temperature a little bit, we can actually fit the data pretty well. We can get a huge reduction in the chi-square, meaning a good improvement with comparing to the experiment, and only at a small cost in entropy, meaning that we sort of retain, in this case, we retain about 60% of the frames of the MD, or we've downweighted 40% and upweighted 60%, so that we have sort of on average about 60% of the MD frames left that then improve agreement with the experimental data by this huge entropy cost. And then what we see is that if we want to get even better improvement with experiment, we really need to effectively throw out a lot of the MD frames. So in this case, we sort of designed the system so we knew what the truth should be. This was the gray line over here. So now we can do this kind of reweighting. We can look at the reweighted landscape. This is the blue thing down here. You can see that the marginals are not identical to nature's force field, but they are much closer than the starting point here, even though we only optimized again one or two measurements, I guess the average of X and the average of Y. You can see that the calculated average is per construction closer to the experimental number. This is what we've optimized. But you can see again the populations become much closer because we have a prior that is not perfect, but at least have the minima in the right places. And again, we can test this in many different ways. This is, as I mentioned, a sort of a dummy system, but we've tested this extensively, and this kind of method can be now done relatively easily and robustly on many different types of data to use simulations to fit experimental data. And as I was updating my talk, I actually cut away all the examples of this because I wanted to talk about other things, but I do want to highlight the approach and the software and just give you this one slide with a little bit of detail of this. This is looking at the protein alpha-synuclein that we and many other people have looked at for a number of years, just using small angle X-ray scattering data. So over here on the left, I show you just one way of representing three different ensembles. So these are three different conformational examples, one of them generated with a simple sort of coarse-grained statistical coil model, and then two different MD simulations with two different force fields projected down onto the radius of gyration. You can see that they have quite different distributions of the radius of gyration. And if you then calculate what you would expect from a sex experiment, actually none of them fit the sex data perfectly. Some of them fit the sex data better than the others. So this is sort of indicated here in the middle. The gray line is some nice SAX data that Lina actually recorded on alpha-synuclein. So these are some good quality SAX data on this. And you can see here's just one example, one of the force fields, that it doesn't actually give you perfect agreement with the SAX data because the compaction is wrong. And then we can do this kind of, Bayesian maximum entropy refinement, and we can essentially reweight it. And now we can reweight it so we fit the SAX data. This is indicated by this blue line over here. So this is the procedure that we went through. We did this with many different models and force fields. I'm just showing you 3 here. And now you can see over here on the right, what is the outcome of this? Well, these are now reweighted ensembles where the frames are the same, but the statistical weights are different. So this is work in particular by Francesco Pesce in one paper, and another paper with Mustafa and Ramon Crevet was also involved in some of this work. And now you can see that, 2 effects. First of all, the radii of distribution, they change. And secondly, even with different priors, or even with initial guesses that are somewhat different, all the solutions that you get out are sort of more similar to each other because we've all sort of fitted them to a single coherent set of experimental data that comes from nature that is supposedly giving us the correct answer, but only seen from one angle, in this case from the angle of small angle X-ray scattering. And then of course we can validate it with other kinds of experimental data, and this is again described in some of these papers. So in this case we're using the simulations not as a prediction tool, but as a tool to interpret the SAX data to get information about, for example, what is the level of compaction and how does the compaction change as you perturb the system. And there are many things that you could do with these things. And again, here's just some snapshots of things that we have done with these methods in different papers, different systems. There are some proteins, some RNA, some big systems, some small systems, NMR data, X-ray diffraction, neutron scattering, X-ray scattering, all types of different types of experiments that we can interpret using these kind of tools, sometimes with coarse-grained models, sometimes with all-atom models, and sometimes with enhanced sampling. And all of these things can be relatively easily plugged into this framework because, in contrast to the sort of top path with the bias sampling, which has some advantages, this method is pretty easy because you just use your normal MD framework. You do your normal sampling. You can use enhanced sampling with your favorite enhanced sampling method, and you plug in the experimental data at the end in our hands, at least, a relatively robust approach. So this was, I just wanted to mention this because I think this is very useful in practical work for many people. And actually, we use this method in these 2 papers that I'll now mention, but actually what I'll talk about is actually not work where we use this reweighting approach, but actually some insights we got from this work that led us to do the next project. The first project was a project on a 3-domain protein called TIA1, or it's a protein where we studied these 3 domains, in particular because there was nice X-ray and neutron scattering data on this, and this is a collaboration with Lisa Alette, and I'll talk about what we did in a second. And the other protein is a project on HNR and PA1. Also has 2 domains, and some of the experimental data also had a fusion sumo tag on it, and then it also has this long disordered tail that is important for the function and also pathology of HNR and PA1. And this is a collaboration with Tanya Mittag's group, and the experiments that were important for our part of it were done by Eric Martin, and the simulation on our side was done by Emil Thomasen. And so we were working on both of these, and we're interested in the sort of large-scale structural properties of these. So we're using coarse-grained simulations with a model called Martini that some of you may be familiar with. And what we found when we started simulating these with Martini and we started to compare with the SAX data is we found that the structures we were getting out of the simulation were all quite a bit too compact. So I'm just projecting it down here now to the radius of gyration. The radius of gyration is 28 angstroms, in this case, and 34 in this case. And you can see the numbers are about 25% too small in simulations, and these are good experimental data and converged simulations. So they suggested that the force fields had some problems, and there are details in this paper describing some sort of ad hoc things that we did to these things. But it also made us realize that probably there is some problem in this Martini model when you want to simulate these kind of flexible systems, and they were not really part of the initial testing and benchmarking of these things. And so what we've more recently done, and this is work in particular spearheaded again by Emil Thomasen, but also with help from Francesco and Mette and Giulio, is that we looked at Martini in a much broader sense with both intrinsically disordered proteins, multi-domain proteins, and also other properties. There's this preprint out. It was actually just updated yesterday or today with additional data and tests. So I'll just give you a few snapshots. But essentially what we did is we collected a bunch of different systems that have good experimental data, in particular, sex data. And then we tried to first benchmark Martini and then update Martini a little bit. And so what you can see is that if you run simulations of, for example, in this case, all these intrinsically disordered proteins, calculate the radius of gyration, you get a nice correlation to experiment, but you can see consistently the simulations underestimate the radius of gyration by quite a bit, showing that the protein is too sticky or too complex. So there's some issue with the force field to simulate these things. And so what we did was inspired by work from other people, in particular Robert Best and G.T. Mittal and many other people in all atom world, is we say, okay, if the proteins are too complex, let's swell them a little bit by increasing the strength of the interactions with water. And I'll tell you about other things that we can do in a little bit, but this is sort of a simple correction. And what we essentially scale the interactions between protein and water by a factor lambda. And what we find is that if we increase this this interaction by about 10%, we get nice quantitative agreement with, for example, the radial gyration from that we get, or slightly more correctly, we can also compare directly to the SAX data. This is sort of the average over a bunch of different proteins, and you can see that you essentially drop, this is a log scale, you drop the chi-square by quite a bit by looking at these IDPs with different behaviors for different proteins. This is discussed extensively in the paper, and also not showing you, but also we looked at these multi-domain proteins, and again you can see substantial improvement in the chi-squares agreement with experimental data. And you will also notice, if you are if you keep an eye on this, right, that the optimum on this kind of systems is sort of different from the optimum of these kind of distance. So there's a lot of things we don't understand, but certainly improving, increasing the strength between protein and water, for example, by 10% here, makes the agreement better for all of these proteins and makes the agreement better for all of these proteins, right? So all of these numbers here are better than the starting point of the force fields. So we also expanded this to many other tests. So for example, we looked at intrinsically disordered proteins, not by sex data, but by various NMR data. Here's data for 5 different proteins. Blue points is the plain force field, and these orange and green are the optimized force field. You can see for this protein, there's a big drop. For this protein, there's a small drop. For these ones, there's a drop. So generally, there's one system here where actually doing this correction actually makes the result worse. So it's not a solution for everything, but it certainly makes things a lot better. We take a bunch of proteins and put 2 proteins in a box. We simulate them together under conditions where the proteins should not interact with each other. And you just take the standard Martini force fields. You can see these things, they stick together, 50% of the time or more for some of these proteins, some of them a little bit low, but definitely a lot more than they should under these experimental conditions. Whereas if you do this, kind of correction, you see that they are essentially, don't see each other much in the experiment. The same is true if you look at dimerization of folded proteins. Again, ubiquitin is known to dimerize, but dimerizes probably too much in plain Martini and around the right number expected, from experiment, and the same thing for Villin. And we also looked at dimerization of IDPs. So overall, this is an improvement. It's not perfect, but it's certainly better than doing nothing. And the details are sort of discussed, in this paper. So again, this all comes out from comparing the simulations directly with experimental data, seeing that there are some discrepancies, so a benchmark, and then trying to come up with some sort of correction that improves the system. But of course, we selected out one parameter from a big force field, the interaction between all atoms and water, and say, can we manipulate this, one parameter to improve agreement? And we could. Not perfectly, but a lot better than we started. But of course, this is a little bit ad hoc in the sense that maybe the problem with this coarse-grained force field is something else, and we should have changed the properties of water, or we should have changed the charges on the coarse-grained particles or something else. And of course, this is completely valid. So it's sort of a little bit of a hack. It's a useful hack, but it is sort of a hack. And so what I'll talk about here in the last part, and I'll just keep an eye on the time here. Is an approach that is a little bit more systematic, or actually a lot more systematic, for optimizing force fields more generally against experimental data, applied to also coarse-grained modeling, and even more coarse-grained model but where we really try to optimize not the entire force field, but many more parameters of the force field. So this work that I'll talk about is in particular spearheaded by Giulio here, but also actually work that started more than 10 years ago by someone else in the group, but it was really restarted again when Ramon was on a sabbatical here in Copenhagen, and Tia at the time was an undergraduate student working on this project. So this is again work from many people that we published very recently, and I'll talk mostly about the published work with a couple of new things at the end. So the idea is that we have again now a coarse grain mod. In this case, it's a 1 bead per residue, and we start with one coarse grain mod that I'll show you is okay but not great, and then we essentially try to optimize more or less the entire force field against experimental data. So we, instead of doing a bottom-up parameterization where you try to target some kind of more detailed model, we really try to optimize the force field directly against the experimental data. So we do this on experimental data on individual proteins in dilute solution. And then one of the reasons why we were working on this was because we're interested in the process of formation of biomolecular condensates. And so we can take this model, we can then look at the equilibrium between these kind of single or dilute phase properties and condensates formed by many of these, in this case, intrinsically disordered proteins, aggregating or forming clusters together of different sizes and affinities. So we started with this model that is called the HPS model. The model that we use is not exactly the same, but it's extremely similar to one described by G.T. Mittal and Rob Best in several papers and their groups. This is sort of one of them. And essentially, each amino acid has a stickiness parameter that is called lambda that ranges from about 0 to about 1. And the interaction between two amino acids comes from just a simple combining rule of these things. So these are sort of sticky residues, and these are not sticky residues, or in another language, they're sort of spacers. And you can see that the hydrophobic amino acids are in this model very sticky, and the charged amino acids, in particular arginine, is very unsticky in these things. Right? And so when you simulate this, you get interactions between these type of residues and looping forming structures of these kind of residues. So this is sort of where we started. But what we also realized was that this model is just one of many possible choices you could have had for how sticky or interaction prone or hydrophobic amino acids could be. So what Giulio did is he simply went to the literature and found a large number on the order of 100 different hydrophobicity or stickiness scales of proteins, and created sort of a probability distribution over these that we will then use as a statistical prior for our optimization. So again, this is now the stickiness parameter and the different amino acids. The blue is the same as before. There's another model that was published sort of around the same time as our paper. That is this orange line here. That is another hydrophobicity model. And then this orange line here is sort of the average over these 100 different, or 80 different hydrophobicity parameters. And then this color here is sort of the distribution of hydrophobicities that we will then use as sort of as a regularizer in optimizing the actual lambda values in this force field in an approach that I'll tell you about in a second. So before that, I'll just show you some sort of baseline evaluation, both with this HPS model and this average model. So if you take this HPS model and you run MD simulations of a bunch of different proteins, you would see that all these variants of a single protein called A1, it actually gets the radius of gyration correct. So these bars show the how accuracy the radii of gyration is, with 0 being good and negative being too compact and positive being too expanded. So actually get the radius of gyration of these correct. But if you take all these other proteins that are more expanded, this model actually compactifies them quite a bit too much. On the other hand, if you take this model over here, you get sort of the reverse situation. It gets all these sort of more hydrophilic proteins more or less correct, but all of these more compact A1 variants, it actually predicts to be too expanded. So this model is okay for these proteins and this model is okay for these proteins, but they're actually not great for the total data set. So what we then did was that we went back to this work that I mentioned more or less 10 years ago and sort of revisited again with the work from Ramon and Tia, and came up with sort of this iterative approach where we want to optimize these lambda parameters. We have sort of a target function that we want to minimize, essentially agreement with the radii gyration from SACS, agreement with NMR paramagnetic relaxation enhancement data, and then also not deviate too much from our statistical prior, which is this distribution of hydrophobicity parameters. So we say we want to stay within the realm of what's known, but we can deviate from it if the experimental data tells us what to do. And we came up with an approach to do this efficiently, because of course, each time we want to calculate these properties, it's an expensive simulation. So we run MD simulation of all of these systems with some particular force field. And then we can do reweighting, statistical reweighting without resampling, sort of with Boltzmann reweighting effectively to optimize the parameters against the experimental data. We can do that locally, but of course, because of exponential weights, we can't do that infinitely far. So once in a while, we have to resample and then do this process over and over again, but optimize the force field to get agreement with a bunch of different experimental data on the proteins that I'll talk about here. So essentially, we have radii of gyration of a relatively large set of different proteins, or of variants of a single protein. You have some fragments of tau. We have all these variants of H9P1 and a bunch of other proteins that are completely different in length and sequence. And then we also have NMR data on some proteins, and for some proteins we have both NMR and SAX data. So we have a relatively large data set, and we're optimizing effectively the stickiness parameters against all of this data. And so how does this look? So in a second, I'll show you a movie of how the optimization works. I'll just tell you what to look for at the end. These are the amino acids. This is the starting point of the optimization, with arginine being very anti-sticky and phenylalanine being very sticky. And if you run simulation, in this case of alpha-synuclein, you can see that the model predicts too strong PIEs, meaning that you have too many interactions in alpha-synuclein, and also the average radius of gyration here is much smaller than the experimental number. And then, during the optimization, we try to optimize the agreement with the NMR data and the SAC data, but not just for these two data points, but essentially for this large big data set before. And so this is the structure that you'll get here. So you can see now, the parameters sort of moving up and down, and you can see we get improved agreement with the PIE data and the SAC data. And then you can see at the end, you get some force field that agrees, with the radius duration, agrees with the PIs, and where the force field is now quite different. So for example, you can see that proline, has dropped quite a bit. These hydrophobics dropped somewhat, and in particular, you can see that arginine went from being extremely anti-sticky to being actually one of the more sticky amino acids, which is actually one of the things that we suspected, because arginine is actually a strange, interesting amino acid that is actually quite sticky. And so we essentially can go through this, this is, a 30-second movie, but it's a quite elaborate scheme of optimizing essentially the entire force field by this kind of iterative procedure of changing the parameters, reweighting, running new simulations, and iterating over and over again to target this large number of data points. So not surprising, of course, once we've done that, we get, we have these three different models that we call M1, M2, and M3. It doesn't really matter how they differ, but, not surprisingly, because this is what we optimize to do, we now get good agreement with the radius of gyration. Again, this is sort of the starting point model that, really predicts many of these things to be too compact. And again, over here, we have all these variants of A1. And again, you can see we get a pretty good correlation between the experimental range of radii gyration. So these are all proteins of exactly the same length. And so the only reason why they get different levels of compaction is because the scaling exponent or the intramolecular interactions are different in all these variants. And you can see we can sort of predict this with not completely perfect, but also, I would say, reasonable accuracy, because we know the relative interaction strength of the amino acids. And you can see we have these different models that I mentioned, and they're quite different from certainly from the starting model. It's quite different from this average model, and it's also somewhat different from this other model that was published by Gita and Mattel. But you can see in particular, it agrees with the fact that arginine is a lot more sticky than what you would normally think. And there's a lot more detail about this in the paper. We didn't write it in the paper, but we've since come up with a nice name for this model. It's called Calvados, in the spirit of naming coarse grain models and force fields after drinks. And this is the name, and this is the reason why it has this name here. So this is now the Calvados force field coarse grain model. One thing I do want to say is that the force field is actually relatively robustly determined. So if you optimize starting either from one point or from another point in parameter space, you actually converge more or less to the same force field at the end. These are the blue and the red points. They're not exactly the same because this was a quite complicated optimization problem, but essentially the combination of the prior and this relatively large data set more or less uniquely determine the force field. Of course, there's some hyperparameters that do change the results a little bit, and the data, of course, do change it. But once we have the data and the prior and the hyperparameters, essentially we can optimize this to more or less a unique solution. Then we can validate it, and there's a bunch of details in the paper. I'll just show you one example here where you can measure interactions between two proteins. So you can label one of them with a spin label, but make it invisible in NMR. And then you can mix it with a protein that doesn't have a spin label, but that you can see in NMR. So in this case, the NMR experiment only probes interactions between two chains when they see each other. And we can show that this model actually captures these intermolecular PREs quite accurately. Or we could look at, similar as I discussed in the Martini case, we can throw two proteins in a chain, in a box, and we can look at the fraction of dimer form. And again, these are 5 proteins that, if you simulate them in the original HPS model, they would stick very strongly together, because overall, for most of these proteins, it's sort of overestimating, at least, in our implementation, the dimer formation, whereas these proteins that should really not form any dimer also don't form any dimer in the simulations, whereas these two proteins that are known to be interaction prone, they form dimers to a small but certainly measurable extent, in agreement with what we would expect from the properties of these cases. And there's other validation that we've done both in the paper and unpublished work also. But the reason why we did this was not just because we cared about the individual monomeric forms of these proteins. The reason why we and other people have been developing these coarse-grained models is we care about how these things can form these clusters via various processes, but in this in particular via liquid-liquid phase separation. And so we can again borrow ideas from other people and simulate, for example, phase separation of these things. We do this in a slab geometry, sort of to make the calculation a little bit more feasible. So we have here a condensate with a high concentration that's in equilibrium with a dilute phase with a low concentration, and we can sample these things. You can see here, this is as time progresses, we can see sort of molecules going in and out of this high concentration condensate, and we can calculate now the average concentration in the dilute phase and in the dense phase. And we can sort of converge these. This is not completely trivial to converge, but we can definitely convert these and get, for example, estimates of the dilute phase or the saturation concentration in a phase separation that we can then benchmark and compare with experimental data. Remembering that we've never looked at this kind of data in the optimization, we only optimized it against single chain properties. But what I didn't tell you, if you work in the field of polymers but also proteins, is that of course there is a sort of symmetry between the inter and the intramolecular interactions. So proteins that like to stick together as monomers and form compact states who have low radioactive duration also like in general to interact with others of the same type, and so that means that they're more prone to form these condensates. And so by training the force field on the dilute phase or single chain properties, we can then hopefully, and this is what I'll show you the next slide, also transfer into looking at condensed phases. And this is again shown here, where we now look at for example, the agreement between the saturation concentration on a log plane. You can see we have a really nice correlation between what we predict from the simulations of these kind of phase behavior and what you measure across several orders of magnitude of changes in dilute phase concentration. We can actually predict this pretty well with this kind of model, because we've optimized it to have the intermolecular interactions correctly, but then this also transfers into interactions between chains. So this we can then now to start to look at this in more detail. This is showing the same concept, but now we can quantify this a little bit more. We are here showing you contact maps, or rather interaction energy maps of the same protein, in this case, the low complexity domain of a protein called fused in sarcona. And so this is a contact map with sort of the white spots, meaning regions of strong interactions or low negative interaction energy. This is the interactions within a single chain. These are the interactions between two chain simulations of a stock with just two chains. And this is now the interactions between the chains in a bivalent condensate. So essentially the central chain and all the other chains. So you can see sort of the picture is, first of all, somewhat similar, apart from one feature, which of course is that in the single chain, we are dominated at diagonal because of course residues that are local, in sequence of course, tend to interact more strongly with each other, but you see these nice white stripes. These are interaction residues that really like to interact with one another, and you can sort of visually see maybe that the patterns are similar. I'll summarize that a little bit cleaner in the same case. Again, showing you that there is sort of a symmetry between what we see in single chains where we optimize the force field and what you see in the condensates. And of course, this is not our idea. This has been known for homopolymers for a long period of time and has been demonstrated both computationally and experimentally by several other people. In particular, the work from Tanya Mitaka and Rohit Papu on the A1 system has shown this very, very nicely experimentally and computationally. So this is the same kind of data now for two proteins, for the same protein, the LCD of Houston sarcoma and the LCD of A2. You can sort of see the similarity. If we just sum up these interaction maps across the sequence, this is sort of the same maps for the one chain, the two chain, and the multi-chain, and you can see there's these dips around the residues that we would then think these are the sticky residues that are driving either the compaction of the single chain or the interactions in the condensate. And you can see that they're in particular, the aromatic residues and the arginines. These are the ones with high lambda parameters that like to stick together, not just with one specific residue, but with many other residues along the chain. And so you can see that this kind of symmetry between the interactions within a condensate and the single chain. There are also some interesting differences, in particular at the edges here, that we're exploring. But overall, there is a similarity of intra and intermolecular interactions, as we would assume, and what we hoped, of course, to be able to see with this kind of model. We can also look at the structural properties of the proteins in the dilute and the dense phase. And again, coming back sort of to ideas coming back from Flory, we see that if we have a condensate where each chain is surrounded by many other chains at the same time, this sort of becomes closer to a theta solvent. And so if you have a protein that has a scaling exponent that's less than 0.5, so it's more compact, it sort of doesn't like the environment, the water in this case, then once it goes into the melt of similar chains, it sort of expands. So we get always sort of a scaling exponent that's closer to 0.5, in this case, exactly 0.5 in the condensed phase, whereas in the dilute phase, all of these are more compact. So again, this is something that you would expect from theory, but also nice to see from the simulation. It's something that's actually quite difficult to measure experimentally because it's difficult to do structural biology inside the condensates. And so you can't easily do scattering experiments, for example, of individual chains in a condensate, but it's nice to see this with the simulations. There's also other details that I'll just skip here about the variance. So just one thing that we sort of realized quite a bit by chance, but actually has made a bunch of our life easier and also taught us various things about these condensates is actually that the long-range non-electrostatic interactions actually have a big effect on the phase properties, but not much on the monomers. So it sort of breaks this symmetry that I was just talking about. And so we are talking about long-range interactions that are sort of the normal Lennard-Jones interactions, not the electrostatic interactions. This is sort of the interaction potential that we are looking at here. And we've simulated this with a very long cutoff of about 4 nanometers. I'm showing it just up here until 2 nanometers. So we're doing cutoffs in force space. So, but these I'm showing you in interaction energy space. So they're almost similar. But if you zoom in on the last part, you can see that there's a difference of just a few joule per mole on a scale that goes on kilojoules per mole. So it's really a small difference here. But what we realized for reasons that I won't bore you with actually is that the simulations actually are quite different depending on whether you use a long or short cutoff. So up until recently, we actually used this long cutoff because, of course, intrinsically you would think that using a long cutoff is correct. But of course, the functional form of these potentials are always a little bit arbitrary in coarse-grained models. So it's not completely obvious that you would expect that one or the other to be more correct. So if we vary the cutoff and we look at just a single chain, you can see that the radius of gyration changes by a few percent, maybe up to in this case these maybe 3%, in this case maybe up to 5-6%. But it's a relatively small change in the compaction as you increase more and more interactions, as you make the cutoff larger and larger, or the other way around, if you make the cutoff smaller and smaller, the protein expands a little bit because you're ignoring some of the outside contact. So this is essentially the data that we've been optimizing against. However, when we look at the actual phase properties, we see the same trend, but instead of having a change of a few percent, we see changes of the saturation content by orders of magnitude. So this is a log scale and this is a linear scale, right? So the dilute phase concentrations are actually changing by, maybe up to a factor of, up to a factor of 50 or 100 as we're changing the cutoff, whereas this one is changing by a few percent over here. And this is, in hindsight, not surprising, because if you have some residue sitting here in the middle of a sphere, if you go out to, to 2 or to 4 nanometers, yeah, there's a little bit of extra interactions that you are sort of including. But if you're in a condensate, there's not just the single chain, but there's all these other chains that it's interacting with. So even if it's only a few joules per mole for each interaction, of course, there are many more interactions in the condensate and in the dilute phase. And so the equilibrium between the two phases actually changed by quite a bit. And this is what's illustrated by these changes over here. And so there's symmetry between single chain and multi-chain behavior in the sense that the RG and the scaling exponents and the saturation concentrations are correlated, but the slope of this correlation actually is also dependent on this cutoff here that is more or less invisible over here. So what we then find, and I think this is shown here, is that if you just take this model that I showed you earlier and you simulate it with a 2 nanometer cutoff instead of a 4 nanometer cutoff, actually all the correlation plots are the same, but all the temperatures shift. So whereas our previous model was really good at capturing the correlations, we had, for reasons that we didn't really understand, we needed to run all the simulations at a slightly higher temperature. And we now understand that this is because there's this imbalance, between the long-range interactions that we can now correct for in a way we understand a little bit better. So now we can actually run the simulation at the correct physical temperature. So we're essentially capturing predictions on an absolute temperature scale, both for these A1 variants, salt effects, these kind of things. The correlations are more or less the same, but we are now able to run the simulations at the correct temperature. And so that means that we can compare more directly with the experimental data. And we are also now optimizing, again, the force field. I'm not showing you that data here. So we're now optimizing the force field with this shorter cutoff and then with a little bit of extra data to come up with a slightly refined version of the Calvados model. But the current model is actually pretty good. And if you change the cutoffs, decrease the cutoffs a little bit, actually you get the right result at the right temperature rather than offset at about 30 degrees. So this is now me at the end with a very brief conclusion on how one can merge experiments and simulations. If you have experiments and simulations that agree, that's great, you can publish your paper. If they don't agree, you can also do a lot of interesting things. You can either do these kind of system-specific corrections that I mentioned very briefly on using this Bayesian maximum entropy method, BME software. Gerhard Humer has some other software that other people have different methods. So use whatever you like to use, but this one is actually, is ours, and it in our hands is pretty easy to use. Or if you have, more data or more patients, you can also actually optimize entire force fields or parts of force fields by tuning them against the experimental data. And I mentioned this work on Martini. So if you like to do coarse-grained modeling of proteins with Martini, I really recommend you to look at this paper and the update now. I really think that it's a substantial improvement with some caveats that are discussed in the paper. Or if you want to go for an even more coarse-grained model, we have this Calvados model that really works pretty well, both for individual chains, but also for multi-chain simulations. And so with that, I'd just like to acknowledge the people that did the work. Not everybody is shown here on this picture, but a lot of the early work on the BME work was done by Sandro Portaro that I showed you earlier, who's not on this picture, and some of the later SACS work was done in particular by another engineer, Francesco Pesce. The Martini work was in particular spearheaded by Emil Thomasen, who's over here with Impact, also by Francesco and Giulio and Mette, who's standing over here. And then the last part on the coarse-grained model was initiated by Ramon and Tia, who are none of them on this picture, and sort of carried through at the end, in particular by Giulio, who's still working on this project. And with that, I'd like to thank all these people for doing all this work and being a fantastic group, and you for your attention, and look forward to questions from you.
Thank you very much. And if you have some questions, yeah, it would be great to either put it in the chat or you could just unmute yourself. So we have a question from Ruyu. Do you want to unmute yourself and ask your question? Or if not, maybe I can just ask his question. So he has two questions. How do you decide how much bias you have to add to reweight the MD ensemble structures to fit the experimental data? And what if the initial experimental data or MD ensemble structures are incorrect? How can you investigate the source of error? So could you look at?
Yeah, these are great questions and something we think about and struggle with quite a bit. So for the first part, I would say that there are automated ways of doing this and they sort of work. In our experience, there's always some choice that you have to make on how much to bias your simulations. And we've tested this with synthetic data in a number of cases, and we have some guidelines to do this. One thing I would upfront say that one thing that we found consistently is that we should not just aim for a reduced chi-square of 1. So a reduced chi-square 1 would mean that we are in perfect control of all the errors, both from the sampling and from the experiment and from the way that we calculate the experimental observables. And this is in general not true. So this temperature-like theta parameter sort of absorbs all these problems, but then because we don't know exactly how well we should fit the data, this is a parameter that we need to optimize. So what we typically do is we look for sort of a kink in this curve that I showed you, and in some cases there's no kink, and then we have to do other things. But essentially by removing, we don't really remove, but downweight a small part of the trajectory, we can do an improved fit. This is a good indication. Then we do two other things. We do cross-validation, and the cross-validation can be one of two different approaches. One approach, which is what we most typically do, is that we leave out some of the data. We try to optimize on one data set and we cross-validate on another data set, and we pick the level of reweighting based on that. And another way that we can do this is an idea that Ramon developed, is that we can do cross-validation, but instead of doing it in data space, we can do it in structure space. So we can optimize the parameters on one set of structures. And because the way that this reweighting approach is done is actually transferable to other structures, we can then we can optimize on one data set and we can apply to another set, and we can sort of see when we start to overfit. So we don't have a perfect approach, but in our hands, with synthetic data and in other type of data, it works pretty well. And what we also find is that if we have to really struggle to reweight, meaning that we have to really get rid of nearly all the MD trajectory, then everything becomes uncertain. So if you have a very bad simulation essentially with no overlap with the real world experiment, then there is that you can do technically things work, but the results become very, very noisy, right? So it's like a one-sided free energy perturbation kind of thing, and then it just becomes quite noisy. And in our hands, the rough rule of thumb is that if we have to go to less than 10% of the original MD simulation, that's left, then everything starts to become quite noisy. But often we can get away with doing much less than that. So this was the first part of the question. There's more detail in several of the papers. The second part is what happens if the experimental data are wrong, and it depends quite a bit on how wrong they are. If they are a little bit wrong, we will just fit them and never realize. But of course, that's where the simulation helps us, right? We are sort of regularizing the fit with the simulation. And so we're trying to minimize the simulation or minimize the deviation from the simulation. And so we will, we will try to stay as close as possible to this prior estimate that comes from the force field. What we found in a number of cases is that we can do the fitting against the experimental data, and we can fit 98% of the experimental data very nicely. And then there's a few data points that we just never can fit. Right? So we improve the agreement to most of the data points, and there's a few data points that we can't fit. And this is one of the nice things then about collaborating directly with experimentalists. Then we can have them go back and look at the data. And in several cases, we've gone back and say, well, we think that this NOE is problematic, or there's some issue over here. And then it turns out that there's often problems with experiments. So when there is clear disagreement with the experiments and simulation, and it cannot be fixed by small adjustments of populations in the MD, this is at least a good reason to go back and look at the experimental data, or in some cases, how we interpret the experimental data. This is very much a case-by-case basis, though I must say that, I think we have a pretty high success rate of finding problems in experimental data with these things. And then one of the nice things about this reweighting approach is that then if there are errors in the experiments and the experimental data sort of changes, we can easily reweight against the new experimental data. So we don't need to run new simulations. We can just change the experimental data and do the reweighting, and it takes 5 minutes or 1 hour, depending on the experimental data. So this is another nice thing about the approach. So this is both good questions and practical problems that there are some guidelines to, but also really knowing the data and collaborating directly helps a lot.
Maybe we could take a question from Ramon.
Hi, Kresten. It's good to know the new slides. I have a question concerning them. Do you think that this cutoff effect is kind of suggesting that maybe the Lennard-Jones that we always take almost for granted is not the best functional form for a coarse grain model, and that there could be better functional forms? Or
I think so, and I think that there would be other so let me step back one system. Of course, intrinsically, I would never assume that using a cutoff would be a short cutoff would be better than a long cutoff, right? If the physics was correct, why should you? I mean, of course, there are corner cases, but this is not what you would expect. So, of course, it's probably pointing to exactly the functional form is wrong. And of course, why would you expect the 12-6 potential to be correct for a coarse grain model? I mean, there's no, neither the 12 nor the 6 has any meaning in this case. So yes, we've actually tried other functional forms that are more complicated that other people have suggested, and they've not made any substantial difference with this. And so at the moment, we just take it as a sort of a phenomenological kind of single parameter that we can tune. To get a good overall scale. But yeah, I mean, absolutely, we can. What we've done is that we've taken our model as it is and refitted the model just each potential to different functional forms and applied that, and that makes no big differences. But of course, the better approach would, of course, be to start with a completely new functional form and go through the whole exercise again. This we haven't done systematically yet, but could do it. The problem being that most of these effects are more or less silent as you look at single chains, and so you don't really have any information about this shift until you go to condensed phase systems. And so we have a new force field that's been optimized with a different cutoff, and it looks very similar to the one with a long cutoff. The main differences are difference in the data and the approach, but it's very, very similar. But all the differences come when you start to look at condensed phase properties. And also what I didn't show, but other people don't know, is that there are other differences between dilute and dense phase related to electrostatics that are also important, because the electrostatic environment in the condensed phase and in the dilute phase, of course, is also very different. So we're using still full long-range electrostatics, but only changing the cutoff on the non-electrostatic sort of Lennard-Jones kind of interactions here. But yes, I agree. It's probably the functional form, but what correct functional form to use is, who knows what that should be. I don't know, at least.
I think maybe in the interest of time, a very quick answer to the question in the chat is that if the arginines are sticky, are they also sticky in folded proteins, and what are the implications of that?
It's a good question. Arginine is a very interesting amino acid, I would say. And I don't have a brief answer to this question here. It is certainly highly charged, so it can form a lot of repulsion, but arginine can also interact with aromatic residues. Arginine can interact with itself. And the more flexibility you have in the interactions, I think this more can be true. So in folded proteins, it's probably less relevant aspect. There are clear differences. So if you do arginine to lysine mutations, proteins change quite a bit. But I think the effects are much bigger for these kind of interactions in disordered proteins. But arginine is certainly not just a simple charged polar amino acid. It can do many things that are complicated.
Yes. I think I would have to agree that I also know examples of these arginines contacting each other and forming salt, which is. Yes, thank you very much.
Thank you.
So thank you very much again for this very nice presentation. And I think in the interest of time, we want to end here. Let's thank again Kresten. And yes, I think we can stop the recording now.
Great. Well, thanks everybody for coming.